So I'm excited to be introducing today's speaker, Dr. Ramakrishnan, who's a faculty member at the National Center for Biological Sciences in Bangalore, India. She pursued her PhD at University of California, San Diego, and her postdoc at Stanford before joining NCBS as a faculty member in 2005. Her research program lies at the interface of population genetics and conservation biology. In particular, she's worked extensively studying tiger populations in Asia. The newspaper First Post has profiled her as being synonymous with tiger conservation in India. Uma is a member of the Indian National Academy of Sciences and has been awarded the prestigious Parker Gentry Award by the Field Museum in Chicago in 2016. I had the opportunity to interact with Uma as well as some of her grad students and postdocs at a conference in India a few years ago. And the resounding sentiment from everyone in her group was that Uma is a fantastic advisor and a great mentor to work with. Today, Uma will be speaking to us about tigers on islands, conservation genetics of small and isolated populations. And I'm looking forward to hearing about her work.
Thanks, Saurabh, for that introduction, and thanks all for coming. It's really nice to be able to share our work with you halfway across the world. So I'll go ahead and start. I'll share my screen, and I'm going to try and go through quite a lot of stuff, various studies trying to tie together different threads. So please feel free to jump in if you have a question. Sometimes it's a burning question and you just like to know the answer. You might be intrigued by this title, the idea of tigers on islands. I guess, as a grad student, I've always been interested in conservation. I've been interested in endangered species, but drawn to population genetics. And I always got a lot of criticism for that in some sense. That how can you think about evolutionary processes in the context of these long-lived species? But I hope to convince you that tigers too are an interesting model to study evolutionary processes. And we do see them kind of playing out in front of us. So tigers, maybe at least some of them, are on islands. At least that's what I feel. Of course, biodiversity is critical to human well-being. We all know this. And the last few years has seen even, public protests across the world, cries for biodiversity and for climate change and so on. Something very different from, for example, several years ago. The cause for biodiversity and biodiversity loss appear very real and something which is going to affect our future. So while this is not a scientific evaluation, some studies suggest that 1 in 4 mammals, for example, are declining and threatened with extinction. So as scientists, we would like to maybe not allow extinction to happen. And we know for a long time, the causes or correlates of extinction. In this case, population size, for example, large populations have low probabilities of extinction, whereas small populations have high probabilities of extinction. And it almost seems like in some cases there's a threshold size. And this has been termed in conservation science, the concept of minimum viable population size. And I very provocatively put this at 100. We'll get to this. This may come up later in the talk, just as a, something as a placeholder. Of course, isolation and fragmentation also correlate with extinction. Some very nice work by Hansky and several of his colleagues looking at how patchiness impacts extinction probabilities. And the more patchy or disconnected populations are, the higher are the chances of extinction. And finally, though this has been debated, hotly debated, and there's a recent paper in PNAS critiquing this idea, genetic diversity is also related to extinction. Populations with low variation are more likely to go extinct. And this is a classic study from several years ago with butterfly metapopulations. But basically, there are direct consequences of small population size, and that is inbreeding and the ensuing inbreeding depression. It's also, of course, in small populations, drift and stochastic events can result in fixation of alleles. And so irrespective of extinction, small populations do have unique evolutionary trajectories, at least in the very least. So I think tigers are a pretty interesting model to study extinction in the wild. And this is in light yellow, kind of a ambitious, a map of their historic range. And basically tigers have lost about 95% of their range. They've also lost populations. There's been local extinction or extirpation. The Caspian tiger, for example, went extinct in the 1930s. The Bali tiger in the 1940s, the Javan tiger in the 1980s. And most recently, the really extensively distributed South China tiger went extinct in the 1990s. Interestingly, tigers from, you can see my cursor, from the Russian Far East are now colonizing China in this really unique kind of a recolonization. But the tigers went extinct, went locally extinct, and also declined in terms of their population sizes for many reasons. But even today, there's ongoing traffic of tiger parts, and this, of course, will result in smaller populations or population decline. So what do we know about tigers from a genetic perspective? So this is some work we just published recently, where we looked at about 65 tiger genomes at varying depths across tiger range. And you can see, for example, that we had a relatively high sample size in certain places like India, because India basically holds 60 to 70% of global tiger populations. We had some sampling gaps, like in Thailand, where we have no wild tigers at all, but we did have wild caught or first generation zoo tigers from the Russian Far East and from Malaysia and captive individuals from Sumatra. So basically, as expected, we find and these are so-called subspecies. So we find, by structure and by PCA segregation, good genetic differentiation of these subspecies. Very interestingly, we find that Indian tigers have the highest genetic variation across all Indian tigers. Nucleotide diversity is high. And even if you look at the number of private alleles, you find that Indian tigers have high private alleles compared to the other subpopulations. Also, interestingly, if you look at this, like these few individuals in this black bar, they're from Northeast India, and they seem to be admixed with individuals from Malaysia. This is where it would have been really nice to have individuals from Thailand. But there is another study which was published a year or so ago. Unfortunately, we were not able to combine all the data in this study, but that study had a few genomes from tigers in Thailand. So now it's possible, now that all these genomes are online, it's possible to combine all these data and actually look at these patterns even a little bit more. We also looked at demographic history, and very interestingly, these SFS-based simulations, which Laurent Escoffier helped us with, revealed that tiger history is quite recent. Divergence between populations is really recent, on the order of 10,000 years or so. This kind of makes sense because in general, tigers are large and they move long distances. So maybe they were relatively continuous until the relatively recent past. Of course, all populations show strong signals of recent population decline and bottlenecks. These are basically divergences within India. And here you see that the northeastern tigers are the most distinct or diverged compared to the other subpopulations within India. But also interestingly, when we look at across all these genomes of tigers across range, when we look at the runs of homozygosity as an index of recent inbreeding with long runs, we find that while, say, the Amur tigers are have very low variation, low genetic variation, they don't appear to be inbred, whereas tigers in India do show, some individuals at least, show signatures of recent inbreeding. So this is kind of may seem contradictory that together all Indian tigers have high genetic variation, high genome-wide variation, but individual Indian tigers are inbred. And I think this is a signature of very high effective size and large areas they occupied in the past, but a lot of recent fragmentation and also very intense recent bottlenecks. But we'll talk about this a bit more. But this was just to position India in the context of tigers. For tiger survival, India is critical because 60 to 70% of global tigers live here, and they also harbor a high proportion of total genomic variation of tigers. But there are already signatures of something, some recent effects on tiger genomes. So if we think about this a little bit more, one way to imagine or visualize what may be happening is to look at how habitat has changed in the past 300 or so years. So this is basically a prediction based on a database called Anthrome, which looks at human population densities and suggests land use types across the Indian subcontinent, 1700, 1800, 1900, and 2000. And you can see that green, the dark green, is pretty much gone, right? So this is why tigers are on islands. They're basically on habitat islands. And, basically protected areas are only 5% of India's land area. And all of these tigers, the 3,000 or so tigers, live amongst more than 1 billion people, right? So there's very high human population densities, and the areas set aside for tigers, at least, are very small, and they tend to be fragmented. Of course, we also know that there's been historically a lot of hunting. Interestingly, this is a picture from the British Raj, and I think it was in 1889 or something like that. There was a bounty imposed for hunting tigers. They were classified as beasts, dangerous beasts, and you would get a reward for hunting a tiger. So there has been human-induced population decline, apart from habitat fragmentation because of increases in human population density and associated human effects. So basically, what we try to do is actually look at these two issues of population size and connectivity, both drivers of extinction, from an ecological perspective, which is simply to estimate population size and understand connectivity, in some sense, using genetics as a tool to understand these ecological processes, but also more recently, use genetics as a tool to understand evolutionary trajectories of small populations. And I'll try to present a bit of work from both of these dimensions. So what we do is pretty simple. Like, we walk through forests, and as I was telling, I think I was telling Chris, tigers are nice because they also walk on trails in the forest, and they mark territories with fecal samples. So all we have to do is walk on these trails. A typical day of field work is maybe walking 25 to 30 kilometers and picking up fecal samples along the way. And you'll encounter several tiger fecal samples, right? Then we bring these back to the lab, extract DNA, and then either, sometimes we sequence bits of DNA to identify species. Is it a tiger or not? You don't want to be looking at a leopard when you're trying to look at tigers. And then also use various types of markers, in the past microsatellites, but more recently SNP markers, to identify individuals and do all kinds of other stuff. More recently, we've also started using shed hair, and this is really advantageous because you can then sample known individuals. So you can follow an individual at a distance in a car, and after a while, when this individual has left, if you go and you look around, you find a lot of tiger hair, which has kind of fallen off where the individual was sitting. So basically, we wanted to actually test whether, is this really a good way to sample genomic data from tigers, or should we just be using SCAT? And so basically, we did this kind of a methods study, and we showed that, while there's a lot of variability, the amount of tiger data, the reads which map to tiger from hair, is much, much higher than you get from, for example, scat. So fecal samples of tigers have lots of other DNA, which is not tiger DNA. The amount of tiger DNA is very low compared to proportionally compared to other DNA. So in this case, for example, in this study, we just use this method to, for example, we were able to very easily acquire whole mitochondrial genomes and identify, like basically there were these individuals who were of unknown matriline, and we were able to identify for some of them that they belong to a known matriline, and this individual belonged to an unknown matriline in that particular park, suggesting that, maybe this was a disperser from somewhere else, or this individual's mother dispersed or some such thing. But basically the idea is that with these types of data, we can actually try to reconstruct pedigrees. And, I might just mention that in the end. So let's go back to India. So here we are in black are the tiger reserves. There's about 3,000 to 3,500 tigers in India. It's not a large number. And the median population size of any tiger reserve is only 40 to 50 individuals. So basically, by all theoretical predictions, these populations do have high probabilities of extinction unless they are, part of larger metapopulations which are connected by gene flow. And there's only 5 of these populations which have more than 150 individuals. So we're really talking about lots of fragmented small populations. Of course, what are the populations is the first question. And so unfortunately, or fortunately, we have to use descriptive population genetics initially to figure out what is the lay of the land. So basically, using various methods, the whole genomes, and earlier on we did rad-seq studies, we find that there's some isolated populations already, like this one, and I'll talk more about it. This is called Ranthambore Tiger Reserve. It's in Western India. It's it's like the most western population of tigers remaining. Tigers did extend into Afghanistan as well, but they're now extinct there. This large block of central India is actually a connected population. As we just discussed earlier, the Northeast is kind of earlier diverged. And basically southern India is also kind of a separate landscape. So basically, we do have already, we have large, these two, southern India and central India can be thought of as large connected populations, and Ranthambore is already kind of a small isolated population. So what is it that allows connectivity in this large area of central India? So some years ago, we did what's called landscape genetics to understand, are there certain land use types that correlate with connectivity? So basically what we did is just collected a bunch of samples. From across several tiger reserves in central India. And you can see how there's just lots of fragmentation across these reserves. And we were able to get genetic data from about 116 individuals. And then we basically inferred resistance or resistance to movement in this landscape. When we map that resistance onto the map, so how do we infer resistance? So basically we assume we have all of these populations and we assume that basically they are nodes which output current. You may be familiar with these models called circuitscape. So these nodes are populations which output current, and the current flows from node to node, and there's resistance between nodes. Now the resistance is afforded by the land use types or the parcels of different types of landscape which are between nodes. You basically assume that the genetic data represents the truth, and you do optimization to see how do you, given a certain type of land use and its resistance, how do you get the best correlation between resistance and genetic data? Okay, so it's a correlation analysis entirely. It's basically trying to infer resistance of specific land use types, assuming the genetic data is giving you a true reflection of connectivity. So for example, when you look at this, you can basically see the high resistance is basically the dark colors, and that is basically roads and cities. Okay. So what we found is that disproportionately, by 2 or 3 orders of magnitude, high traffic roads and high density human settlements afforded high resistance. It didn't matter too much whether you're talking about agriculture or forest. It wasn't it was not, agriculture was not that detrimental to tiger connectivity, right? But high traffic roads and high density human settlements were. So yeah, so I mean, this is interesting, but it's kind of, maybe it's a little obvious. What is not so obvious, and what we were very curious about, is the landscape is changing, and how is that going to affect the future connectivity of tigers, assuming that our inferences about connectivity and land use are correct. So we basically modeled the future. Of course, we don't know what the future is, but we modeled 56 scenarios or versions of the future. We had kind of classic no change at all. We constrained the forest area, we constrained croplands. Business as usual was a scenario where we looked at change over the last 10 years in terms of how much forest had been lost, how much agriculture had increased, and so on. And we just extended that into the future. These simulations were 100 years into the future. And then we also allowed for this very extreme scenario of fenced protected areas. And I'll talk a little bit about that later, because when tigers are moving through these landscapes, there is conflict with humans and livestock, right? So then we also allowed, for example, tigers to be only inside protected areas, or to kind of bleed through the landscape and live outside protected areas as well. These are individual-based simulations of tigers, landscape, and resistance, right? And what did we find? So at the end of this, 100 years, we look at extinction probability by looking at how many times out of 1,000 simulations or whatever, that particular population goes extinct, right? And here we're looking at those two metrics we talked about, population size and isolation index. So obviously when the isolation index is high, populations are isolated. When it's low, they are connected. So, as we expected, small and isolated populations had high probabilities of extinction, and large and connected populations had low probabilities of extinction. And what's interesting is large is not that large. So this population has 60 to 70 individuals, but it seems to be very central in this landscape, driving connectivity and also never going extinct. We had a bunch of populations which were in the middle, with moderate extinction probabilities, and also in this demographic realm of having moderate population size and moderate isolation. So from a perspective of the future, what should be considered is how could we minimize extinction probability for these populations? Should we increase the number of individuals here, or maybe increase connectivity? What might result in a better outcome for any particular population in this landscape? So I think these kinds of scenarios are interesting, because it helps us in a particular population to think of what might be an option for the future. And what was interesting was actually that this population I mentioned to you, Kanha Tiger Reserve, it's right next to Pench, where Rudyard Kipling wrote Jungle Book. And this is actually Pench Tiger Reserve. And right between them is basically, this 2-lane highway, which was a national highway. So we have really narrow highways in India. But then there was a proposal to expand this to a 6-lane highway, which would suggest that there would be a barrier between Kanha and Pench. And basically, because we had done these simulations, we were able to very easily model if this became a barrier, how might that affect extinction probability. And we showed that basically extinction probability increased by 50% in several of the populations. And that was actually used in court, not by us, but by an NGO to petition for an underpass, which was actually approved, because we were actually able to say that, quantitatively, this will be the impact of disrupting gene flow. So though these models may seem simple-minded, it was actually able to inform something on the ground, which today is allowing connectivity to persist in this landscape. So of course, when we have connectivity, that means that tigers are moving between these protected areas. And what that means then is they're moving through human-dominated landscapes, which often results in conflict. And so we've been doing this fun project where we've been trying to figure out more about conflict. And I'll show you this video. This is a tiger which has killed a cattle. It's actually just a camera trap video. And you can see that it's actually licking the cattle. And so that's basically good for us. Every cattle kill is like an investigation site where there are lick marks, there are bite marks, basically the jugular bite mark where the kill actually happens. There's hair associated with eating, and then there's fecal samples which may be around the site of the kill. So basically my student, he's a crazy guy, and he's been tracking these cattle kills around a particular park, and he's distributed these little cards that, if it just says, if a tiger has killed your cattle, call me. And so basically he gets these calls and he rushes to these sites, samples all this material. But the idea is that we can then, so this is just another example of, you can see the lick mark very easily. It's not like it's very ambiguous. We've shown, for example, that there's a difference in DNA concentration between the rainy season when most of the DNA is washed off and the dry season, and also in terms of the number of SNPs typed. Actually, saliva, shed, hair, and scat work relatively similarly in these cases. What's really interesting is to actually see this is that same park we were talking about, Kanha Tiger Reserve. These are all the individuals identified from about 100 kills sampled over a year. And you can see, for example, the different individuals are just represented by numbers. You can see that there are spatial hotspots, locations where several individuals are killing cattle, right? As well as, potentially places where the same individual is killing many cattle, right? So it could be, and this would be even more accentuated, for example, if one individual is making kills all over the place, right? So basically what we're trying to do now is to understand what drives conflict. Is it some kind of a spatial distribution of cattle and, kind of pinch points, edge effects, or is it something which is driven by individual behavior? Are certain tigers more likely to kill cattle habitually than are others? So now I'll switch gears, and that's basically ongoing work. But I'll switch gears, and I put it in also because I met Savannah yesterday, and I thought she might be excited to hear about it. To this other very beautiful, charismatic population of tigers in Ranthambore, which is in Rajasthan. You can see in the background this beautiful pink building. It's an erstwhile hunting lodge. And of course, these hunting lodges are now abandoned. And this is a really hot place. And now tigers often rest in these hunting lodges because they're really cool in the summer sometimes. So Ranthambore is this like this National Geographic population? Everybody in the world comes to see tigers in Ranthambore. But it's actually small and isolated. And from a perspective of a population geneticist, the burning question is, are tigers here inbred? Do they actually have a higher mutation load? Or is there evidence of purging? We don't see any, I should say right off the bat, we don't see any abnormalities, at least very obvious ones as of now. So basically, we are in some sense lucky here because this particular population, a lot of times tigers are caught because whatever, they moved or something like that. So we were able to actually get blood samples from several individuals. This is actually really something very hard. And we were able to sequence about 18 individuals from here and a total of 57 genomes, whole genomes from across India. And basically, we use this framework of small isolated versus large connected. And so we have the large connected Central Indian population, and we have the large connected Southern Indian population. Basically, this is not connected to other populations, but there are several tiger populations here which are connected to each other. Whereas if you look at Ranthambore, it's a single isolated population. There are two populations here, but basically this one, it's called Sariska, and tigers went extinct here, and all individuals here are introduced from Ranthambore. So they're part of the same pedigree. Basically, they're part of the same family. And basically, we have not quantified inbreeding through pedigrees, but we are using ROH, or runs of homozygosity, as a proxy to look at inbreeding. And you're all very familiar with the fact that, because of inbreeding, you tend to inherit chunks which are basically identical by descent. The longer the chunks, the more recent the inbreeding. So basically, when we look at all of these different populations, small isolated, large connected, and two large connected, one in the south and one in central India. And if you look at FROH, where we are assuming we are counting all bits which are longer than 1 MB, we find that disproportionately tigers in Ranthambore are have very high FROH. On an average, they're twice as inbred as tigers in other parts of India. This is also reflected in the recent effective size. So this is basically effective size of these three populations calculated based on ROH and assumed mutation rates. And you find, recombination rates, you find that in general, effective sizes are lower, but in the past, they were relatively similar. What's also interesting is not just that there's high runs of homozygosity, but individuals in the small isolated population share these runs of homozygosity. So this is the proportion of shared IDB stretches. They share almost 40% of these runs with each other. And for example, we can compare here to zoo individuals where we know what inbreeding coefficients are. So not as bad as them, but definitely quite a lot of shared stretches. So we can then also predict loss of function and missense mutation alleles. And we can look at how the number of damaging homozygotes varies as a function of FROH. So basically, as expected, the more inbred an individual is, irrespective of population, irrespective of which population it belongs to, there are more damaging alleles. This effect seems to be a little, is less strong with the loss of function alleles. So this is basically, you can see that this is pretty much linear, right? But this is not. So basically, there seems to be fewer loss of function alleles in the small isolated populations than in the large connected populations for individuals of the same F, right? So basically, this suggests that, well, also because these are in homozygous state, so these individuals are possibly still prone to inbreeding depression, possible fitness effects. Why? Because these are putative loss of function or damaging alleles. So we can also look at relative mutation load by this basically contrasting mutation load for loss of function, missense, and intergenic mutations, loci. And what you see is again what we just saw earlier, but basically for loss of function alleles, actually the small isolated population has relatively fewer than either the large connected population in South India or Central India. So this is basically, as we understand it, evidence of purging of the loss of function alleles in the small and isolated population. However, when you look at missense mutations, that's not the case. We are assuming that fitness effects here are probably much smaller, and maybe that is the reason for this. So what we find is a kind of an interesting combination of two things. that, people usually try to say, oh, it's either purging or inbreeding depression, but we seem to find potential for inbreeding depression, but also for also evidence for purging. So while it seems like several loss of function alleles are purged, there are more missense mutations in the small and isolated population. We can also look just at the distributions of these alleles, right? And basically what we find is across the board, there seem to be fewer neutral alleles than loss of function alleles in the lowest allele frequency bins. Okay, again suggestive of purging, but this is even for all Indian tigers. So this seems to be pervasive across Indian tigers, not only for the small and isolated populations. And this is probably because of the founding events and the bottlenecks which have happened in the last 200 or so years, kind of, sort of around the time of bounty hunting onwards, right? But this is something which we probably need to explore more and see what exactly is happening. So I hope I was able to communicate this without it becoming too confusing. There are these long homozygous stretches which indicate in the small and isolated population that individuals are inbred. Also, individuals share these inbred stretches, and these inbred stretches harbor obviously homozygous because they're ROH and damaging alleles. So that sets up a scenario for possible inbreeding depression should these predicted damaging alleles have fitness consequences. There's a very nice paper I just saw on bioRxiv, of course, by Josephine Pemberton's group, where they actually have been studying these soy sheep for many, many years, and they actually know the fitness effects of alleles. And so they are actually also showing just now that older ROH stretches have fewer damaging alleles, suggestive of purging, than younger ROH stretches, which makes sense, but it's actually really nice to see that. So of course, the other thing though is that all Indian tigers show evidence of purging, and basically that there are less damaging alleles in the low frequency bins. But while there is purging, it's not like all damaging and loss of function alleles are purged from these small populations. So we're not saying that there's never going to be inbreeding depression here. It's something which would be important to kind of follow up on. So what we're trying to do is, of course, get some ideas about fitness. Very difficult to do in the context of carnivores, but we're basically attempting to reconstruct the pedigree with these, identified individuals and the hair and so on. So right now we have about 40 individuals out of 60 or so, which we have sampled for hair, and we'll see how that kind of pans out. So now I'll tell you another story. How am I doing on time? Okay, I guess okay. Of another isolated population, and I have to confess, this is completely like an exploratory story. But it's fun to do science that way as well. So there's this park called Simlipal Tiger Reserve over here in eastern India, in the state of Odisha. And what's really cool about Simlipal is it's the only place where these pseudo-melanistic tigers have been camera trapped. So why do I say pseudo-melanistic? They're not completely black, right? Like melanistic leopards. They have stripes, but it's like someone dropped paint on them, and they're like all the stripes are array. They're not regular, right? And so these pseudo-melanistic tigers have been described in the literature, but whatever, they're just descriptions. But they were really not known until actually they were camera trapped. This is the camera trap over here. It's basically, you put it at that height, and basically there's an infrared beam. When the tigers cross, it clicks pictures. So basically when these tigers were camera trapped, that was the first evidence. I mean, people had said they've seen them, but this was the first concrete evidence that such a phenotype existed in only this one location. And, I think that's a pretty good thing that, you can have pretty high confidence in that statement, because India surveys its tigers with camera traps. And there's a huge effort which goes into laying camera traps all over the country. And in fact, this year they went into the Guinness Book of World Records for the largest animal survey of any kind in the world. So there are a lot of camera trap tigers, basically. So when we say they are only seen here, that's a pretty accurate statement. So of course the question is, why do these tigers look like this, and why are they only in Simlipal? So for me, the first thing I could think of was drift, but of course we had to investigate. So luckily for us, there's a bunch of work on tigers and coat color and pattern. So basically, actually on coat color. So this is work by Shujin Luo and her group based on tigers in the US, actually. Basically, this is a white tiger. This is a golden tiger, which is basically like a bleached out version of this. And then this is a snow white tiger, which is a double mutant of this mutation and this one. So this is the mutations for these two phenotypes are known. They are both recessive, and this is basically a double recessive. Also, very interestingly, Chris Kalin and Greg Barsh they've worked for a long time on patterning in cats. And basically, they found this really interesting pattern. This is a tabby cat. This is a marbled kind of cat. And what they found is that there's a strong genetic association between this phenotype and mutations in a gene called TACPEP. So basically, this is when TACPEP is normal, you have during development a regular laying of stripes. But when TACPEP is mutated, you do not have that. And so basically the stripes would go array, right? So luckily for us, we had a candidate gene. It's TACPEP. Most probably these pseudomelanistic tigers have some mutation in TACPEP, okay? But how the hell do we get at that from a wild population of tigers? How do we track down the genetic basis of a phenotype in the wild? So luckily for us, in the zoo close by, at some point, I guess there must have been an individual which came from Simlipal, but there was a litter which had, basically a white mother, a white but melanistic, pseudomelanistic daughter, and an orange pseudomelanistic son. Okay, so we had this pedigree, and so even here we were not able to get blood samples, but we somehow got saliva, whatever, and we were able to sequence genomes from all of these individuals as well as one generation back. So before I go there, I'll just show you this. So basically, it turns out that you can do an association of there's one mutation in the TACPEP gene which is present in recessive state in pseudomelanistic individuals. And when you look at the pedigree, the mutation in the context of this pedigree, you find that there's 100 times higher probability that this mutation causes the phenotype, whereas any other random mutation in the TACPEP gene. Okay, so basically we've identified the mutation which causes this phenotype. Now, it also turns out that Simlipal is small. It's way smaller than Ranthambore. It probably has 15 to 20 individuals. That's it. Very small population, but also in the context of the larger central Indian landscape, it is isolated. Okay, so it's not very well connected. Like, remember, we were talking about Kanha. So Kanha is the one with large variation, and we had looked at landscape connectivity in this area, but Simlipal is basically isolated. Okay. So basically the first question then is, maybe this allele is present in a heterozygous state, and we just haven't seen the phenotype, right? But it's there elsewhere in different parts of the world or in India. And so in order to look at the frequency of this allele elsewhere, we actually were able to, after a lot of effort, this took much more time than some of the other stuff in this study, to genotype this mutation for 417 individuals across tiger range. And these boxes are individuals from zoos. To the best of our knowledge, apart from Simlipal, pseudomelanistic tigers only occur in 2 zoos. And we confirmed that in both those cases, the pseudomelanistic individuals had this mutation. So this is an independent pedigree. Well, I'm sure it's correlated. We don't know for sure, but at least these individuals are not immediately related to these in this zoo. So basically, though, what was kind of crazy was that while we did not find this pseudomelanistic allele or mutation anywhere else, in Simlipal, its frequency was 60%. Okay, so basically 30% of the tigers are pseudomelanistic. So that's 0.6 squared, right? Because it's a recessive phenotype. So outside of Simlipal, it doesn't seem to be there at all. Now, maybe if we had more individuals, you would find it, but still this is quite a large number of individuals to have not found it. So, okay, so then, is it just that the individuals in Simlipal are inbred? Is that why you have a high frequency in Simlipal? Well, they're not as inbred as the individuals in Ranthambore, okay, which we just talked about with the whole genomes. This is, of course, much messier because this is based on non-invasive fecal samples and SNP data from about 150 to 200 markers. But basically, they're definitely more inbred than individuals in, this is pairwise relatedness, but they're more related than individuals in Central India, the large Central India cluster or the Southern India cluster, but not as related as individuals in Ranthambore, which we know is an inbred population, right? But yes, overall, they seem to be quite highly related to each other. So the other question we asked very simply is it possible to have such a large jump in allele frequency from whatever, around 0 to 0.6, and how long might that take? What kind of a bottleneck would you need? So basically, you just need a very intense founding event, and it doesn't really matter how long ago it is. But if you have a founding event of two individuals, one of whom must carry the allele, you can get appreciable probabilities, like 30% chance that you will have, a higher 50% or higher allele frequency in Simlipal today. So basically, I guess we can't rule out selection at this point because we don't have whole genomes for which we could test whether this locus is under selection. But it's possible by founding events and drift that we see, because of a loss of connectivity and small population size, a high local frequency of an otherwise rare allele. And of course, continued isolation would fix this allele. Whether that's bad or not, I don't know, because I don't know whether this has any fitness effects. Probably not. But again, to understand that better, we'd have to do a long-term study where we looked at reproductive success of melanistic versus non-melanistic individuals. So I think I'm done. Just to summarize, tigers in India live in this diverse setting of connected and disconnected, small and smaller populations. I shouldn't say large, since no population is really large, right? There is genomic evidence of inbreeding, but I would say actually that it's not too bad. Even the 100 plus size populations do not have such high mutation load. But overall, we do see evidence of purging in the small and isolated population. There is evidence for drift as well, and founding events. So we are seeing, in some sense, evolution playing out in these small populations already. I'm really keen in the future to work with historical skins. I'd like to, of course, understand more about these ROH and the putative deleterious alleles. I'm not sure how. Maybe using, it's not something I've done, but using tools like CRISPR, or I don't know how. But also, I'd like to see how old these are. So if we go back to skins which are 50 years old, do we see these long runs of homozygosity, and so on. And of course, I'm really keen to understand much better fine scale connectivity and movement. And I actually, I'm not very happy with these landscape genetic models, because I feel like they don't incorporate behavior at all. Um, it's like static, tigers are just moving. But in reality, tigers do make choices. And if we could, I don't know, somehow develop agent-based models or frameworks to understand connectivity from the perspective of individual decision making, that would be really interesting. Of course, we have no data on that. Like we don't have data on, like we would need, say, for that we would need hundreds of radio collared tigers, which we don't have data on. We have data from about 5 radio collared tigers or 10, right? But I wonder whether if we did fine scale genetic sampling, lots and lots and lots of fecal samples, we could actually try and understand movement a little bit better, maybe. So I'll just stop and thank, I've been very lucky to work with some fantastic people. Prachi did the work on connectivity, and Meghna did a lot of the population genetics. Anubhav here is the one who's worked on ROH, and this is our team in Ranthambore Tiger Reserve. We're very lucky often to work with local people who know way more about these animals than we can ever hope to learn. This is Vinay, who's worked on the melanistic tigers. And of course, I've had a lot of funding from many organizations. All of this work is quite expensive, and I'm really lucky to be able to do it. So thanks to all of you. And yeah, just leave you with that picture of Ranthambore and take any questions.
So if people are being shy, I'll start by asking a question. So many years ago, actually, my wife and I had occasion to go to Ranthambore and see and see the tigers. And what was incredible to me, one of the things, aside from the tigers, which were mind-blowing, about Ranthambore, to me, was that it was basically an enclosed area for hundreds of years.
Yeah.
And so I wonder if I wonder if you've done any comparative population genetics across species to look at the generalized effect of that sort of fragmentation. Do you see it in all species? Are there species that you see it into a lesser degree? Are there ecological correlates? For instance, one of the things that was incredible when I was there is we saw a sloth bear. Now, there's no way that sloth bear isn't like some weird fragmented population of sloth bears, right? So have you done any of that work?
Yeah, so basically, we've not done it in Ranthambore, but certainly in central India, we've looked at multi-species connectivity. And there are lots of differences. So sloth bear, for example, are actually much better connected. Leopards are better connected than our tigers. But yeah, so I think that it does depend on the species, on body size, and so on. I think Ranthambore was connected in the past. Definitely it was a hunting area where there's historically been hunting. But it would be nice to actually look at other species also in Rajasthan, maybe, to see what patterns of population structure we see. But yes, probably most of those leopards may not be isolated though. There are these Aravalli mountains, and they are able to move, along those mountain ridges. So the thing about leopards is they live a lot also in human-dominated areas. So for species which are generally dependent, tigers can also eat cattle, but they're much more dependent on wild prey. So for such species, I think isolation will be much stronger. What happens with Ranthambore is there's this big river, which is also a divide. So actually, distance-wise, it's not very far from other tiger reserves. It's just from a genetic perspective, it looks very distinct because all the individuals are related and the allele frequencies are correlated.
I was wondering from like a management perspective, how often are individual tigers translocated between protected areas? Is that something that's done or not really?
All the time. It's a
Okay.
fun thing for the managers to do, no, very macho to like catch a tiger and move it. And they do it sometimes with problem animals, which is a bad thing to do for a cat, because cats, I have a friend who's worked on leopards, and she actually showed that leopards are translocated all the time. And because they go back to their territories, they actually cause a lot more conflict on the way going back to where they're from. So basically, for example, in Ranthambore, other populations of tigers in Rajasthan have been rewilded with Ranthambore tigers. So we have, for example, suggested to the managers like which are the least related, or whatever. But yeah, they are moved all the time. So the thing is that they're moved, but they're not moved based on any science. I mean, they just move like whatever wants to move, whichever animal.
Okay, that's interesting. Thank you.
If other people have questions, you can unmute yourself, or you can raise your hand, or put it in chat.
Great talk. I was wondering, about the temporal sampling with the old skin tissues that you have, do you have a lot of tissue, or have you started working on that? Do you know the quality, and how far back can you go?
Yeah, so we haven't started yet. They're mostly going to be pelts, so like flat cured skins. We might have access to some skulls, so we might take turbiners. They are really good sources of DNA usually. I don't think we can go very far back. So for example, I done an earlier study, which I didn't talk about, where we got skins from the London Museum of Natural History, and the oldest they go back is 200 years. But most collections were done in 1915. There was a British a natural history collection trip. So a lot of collections from India date around that time in international museums. The skins I'm also trying to access are people's private collections, like these kings and stuff like that. So those are probably a bit more recent, but basically between 100, 120, so not very recent. It's not going to be pre-bottleneck, which would have been nice. It's like 500 years ago would have been nice, but I don't think we can get that. And for the methods, sorry.
What is the generation time for?
5 years.
5 years, okay. Yeah.
So not a lot of, so basically one of the challenges is estimating recent demographic history. So anyway, we'll see what we can do, but we're hoping to actually use capture probes and see if we can we'll see, what DNA quality we get. I have no idea. These are all skins in people's houses. So they've not really been maintained at some temperature or anything like that. The other thing which we're kind of interested in looking at is whether there's any, so the striping pattern would be quite interesting to quantify in some way. And there are some individuals which have left-right asymmetry. I've just eyeballed it. It seems to be quite a lot of variation, but it would be really interesting to see whether there's any kind of morphological correlates of these inbreeding coefficients or FROH. There's a lot of pictures. So there's a lot of these people, like, and you went as a tourist. There are so many tourists and there are so many pictures. And one thing which might be interesting is to try to take all these pictures and see if we can get some kind of a morphome, some kind of a, I don't know, many measurements or something like that. We haven't tried it yet.
We're at 10, so maybe we should thank Uma again. Thanks, that was great. And I think, I forget, so